What did the most primitive forms of life look like in the form of the first replicators based on RNA molecules? What are ribozymes? We will try to answer these and similar questions in today's episode of the series Secrets of the Gene, in which I welcome you. My name is Eduard Kalinovsky, and I work as a geneticist at the Institute of Biophysics of the Czech Academy of Sciences. After outlining the possible locations where the most primitive forms of life could have originated, let's try to look at what the first molecules of life might have looked like. In the following section, we will describe the currently dominant concept of the RNA world, which most likely preceded the world of DNA, RNA, and proteins. Current theories place in the most distant times we can observe there was a world of RNA molecules from which the world of RNA and proteins gradually developed, and only later did the DNA molecule emerge. How colorful was the palette of ancient molecules of life? What was their chemical composition? And what was their structure? We will probably never know. Just as with the places where life originated, when it comes to the form of the first molecules, we can only sketch out possible scenarios that are consistent with modern knowledge and therefore, with greater or lesser probability and accuracy, reflect the ancient reality, which in any case is lost in the distant past. When thinking about the origins of life, scientists have often asked themselves, what came first? Was it nucleic acids or proteins? During cell division, DNA molecules are copied so that each daughter cell receives the same genetic information. And copying DNA cannot happen without the presence of proteins as catalysts. On the other hand, the information for building proteins is encoded by nucleic acids. So the question of what came first is a bit of a vicious circle, similar to the classic question of whether the chicken or the egg came first. A breakthrough moment in the search for the oldest molecules carrying hereditary information was the discovery of special nucleic acid molecules with enzymatic functions. They were only discovered in the 1980s of the 20th century, and they are called ribozymes. The name comes from the words ribonucleic acid and enzyme. Please don't confuse them with ribosomes, the protein factories. Ribozymes combine the properties of nucleic acids and enzymes. They are able to store hereditary information and also function as catalysts for reactions. And this was exactly what scientists searching for the first molecules of life needed. Ribozymes were able to join or split other RNA molecules without the help of proteins and even create their own copies. The discovery of ribozymes was so significant that their discoverers, Thomas Czech and Sidney Altman, were awarded the Nobel Prize for it in 1989. The problem was solved. Another Nobel laureate, Walter Gilbert, proposed the existence of an RNA world that preceded today's world of DNA, RNA, and proteins. RNA molecules therefore can both store and execute genetic information. It was only later in the course of evolution that protein synthesis emerged and the role of storing genetic information was taken over by DNA molecules as they were much more suitable for this function. Remnants from the era when RNA molecules dominated have survived to this day in the form of various types of RNA molecules. In the previous episode of the series, we saw that RNA molecules still participate in many cellular processes today. They demonstrate that the transition from the RNA world to the world of DNA and proteins is still ongoing. Moreover, the genomes of all organisms are infested with parasitic segments of DNA. What's interesting about them is that they jump around the genome from place to place using RNA molecules as an intermediate. The alternation of DNA and RNA molecules in their life cycle may reflect a transitional stage and perhaps serves as a kind of reminiscence, a memory of ancient times when hereditary information was carried solely by RNA molecules. Next time we'll talk about why it was evolutionarily advantageous to use DNA molecules to store genetic information. There are more such relics from the RNA world. An example is probably the protein factory, the so-called ribosome. It is made up not only of proteins but also of RNA molecules. Ribosomes have even been created that are made solely of RNA molecules without any proteins, and they did their job. They synthesize proteins. The twist is that for a long time, scientists searched for a protein that would catalyze this important reaction, only to discover that it was actually an RNA molecule, still part of the ribosome, that was responsible. The key reaction in protein formation is the creation of the peptide bond. This is therefore carried out by a ribozyme. What happened to the original ribozymes? Many of them have certainly disappeared. Some retrained for a different function, while others retained their original role. A long time ago, there was a primary division of labor among biological molecules. Proteins took over the role of executing genetic information from ribozymes, distinguished by higher speed, accuracy, and a broader range of catalyzed reactions. DNA then specialized in storing information. Thus, the world of RNA branched out into the world of DNA, RNA, and proteins. That's all for today. In the next episode, we'll talk about the origin and evolution of the genetic code. I'm looking forward to seeing you. If you enjoy my storytelling, I'd appreciate a like. And if you'd like me to continue making episodes about genetics and evolution, please subscribe, which will help these videos reach more people. I really appreciate your support. Thank you, and bye-bye.